Hey guys, Mrs. Hone here. Today I've got some information on different technologies that scientists use in the field of genetics. I'm going to explain each one and tell you how they're currently being used. We're going to cover notes on page 31 through 36. If I go too fast and you need more time to write things down, feel free to pause the video and continue when you're ready. We're going to start on page 31. Talking about recombinant DNA. A piece of recombinant DNA is DNA that comes from two different sources, maybe two different types of organisms. In the example that I'm going to show you, it would be DNA from a human and DNA from a bacteria. In order to make this work, you would need a plasmid, or a piece of DNA, a restriction enzyme, and some DNA ligase, which we've talked about ligase before, and ligase is the enzyme that helps to attach pieces of DNA, kind of glue them back together. I'm still on page 31. You don't have this picture though, but this shows us what DNA looks like at the top. The purpose of a restriction enzyme would be to cut the DNA, kind of cut it open, and allow these, what are called sticky ends, very scientific term, and then those sticky ends will become the combining place for new DNA, whether it be some bacterial DNA or some human DNA. Using this technology, we can take a human gene and put it into bacterial DNA. That bacteria can now produce things for us. For example, we can take the gene for making insulin. We can take that, put it into bacteria cells, and then those bacteria cells can in turn make insulin for us. A bacteria cell does not know how to make insulin on its own, but if we put the instructions in their DNA, then they can make insulin for us. The benefit of this, we could have an unlimited supply of insulin for diabetics to utilize. We could also use this technology for them to make hormones for us, or blood clotting factors for hemophiliacs who can't clot their own blood. Another technology that we use is called polymerase chain reaction, or PCR. This could very well be something that we talk about when we go to the U of R. But the purpose of this procedure is to create many copies of one gene, or maybe a piece of DNA. So we use PCR to copy pieces of DNA. We might do this when there's a crime scene and DNA is left behind. It might only be a small amount, but we want to do many tests on it. So in order for us to get more copies or a larger stock supply of DNA, we put it in a PCR machine, and after a couple of hours, it actually makes copies all on its own. So it's just like DNA replication, which would be a natural process that your cell does before mitosis. But PCR is a machine that geneticists typically have in a lab that they can make copies of DNA to use for additional testing. Maybe the original sample they have wasn't big enough, so they need more copies. And this is the picture that you have on page 32. The original piece of DNA that they had, and then over a few hours, more and more copies of the exact same, all of the exact same DNA are made. So within a relatively short period of time, you go from a small DNA sample to you've got lots of DNA. You can run all kinds of tests. I'm moving over to page 33. This is a process called DNA electrophoresis, or gel electrophoresis. The picture that I have on my slide is a really good representation of what it would look like as an end result. The picture that you have on your page is a good explanation of the steps that I have here. So once you have a DNA sample, you're going to cut that DNA with restriction enzymes. The restriction enzymes will cut the very large piece of DNA into smaller segments. They won't all be the same size though, so some segments will be large and some will be very small. But the whole piece of DNA from one source, maybe one person, would be inserted into one of these gels here. I'm kind of circling one, I'm sorry, into one of these wells, which is step 3. All of these, they look like little holes up here, and those are the wells, and it's like a hole in the gel, and it holds onto the DNA sample. So using an instrument called a micropipetter, you would insert the DNA sample into here. You would then hook this up through electricity, and because DNA has a negative charge to it, the DNA pieces are going to want to move down the gel because this end of the gel is positively charged. So the negatively charged DNA wants to move towards the positive end. Small pieces of DNA travel very far. Big pieces of DNA get stuck and can't move past a certain point. But the really small pieces, the bands, move farther down the gel. So in the end, what you can do is you can see DNA patterns of individuals. So maybe one of these rows is from mom, and one of them is from dad, and some of them are from the different children that they have. So you'd be able to match up and use this for a paternity test, or you could use it to match DNA to different suspects that the DNA may have been found at the crime scene, and you match it up to different suspects. Here's an example of a gel that has DNA from mom and DNA from a child. And any of the bands that match mom have been color-coded purple. So then any of the leftover bands that didn't come from mom have been color-coded red. And then we have 2 possible dads over here. And the one that has the matches is going to be dad. So it looks like dad number 1 is baby's dad. I'm moving over to page 34, and I want to talk about products made through biotechnology. What we have here in this picture is we have found how to take a gene for the human growth hormone. We can then take that gene and insert it into an egg of a goat. That embryo is then implanted into a female goat uterus, and the end product is a goat that produces growth hormone in its own milk. So we have found a way to create an actual organism that will literally give us what we need in its milk. We could take this even a step further, and once we have some goats that make this human growth hormone milk, which by the way, this milk is needed for many different people that don't make growth hormone. Their body doesn't make growth hormone, so if they can drink this milk, then they get the hormone. So it's kind of a good deal. But if we took it one step further, why not just clone all of these sheep so that we have lots of sheep that can produce this type of milk? So this is a technology that is most definitely a possibility. Something that we've tried, we have what we call a spider goat. This information could go on page 34. I don't want you to write the whole thing, but if you could summarize this in a few sentences, that way you'll be able to remember we talked about the spider goat. The spider goat, they have taken a gene from spiders, the gene that tells the cell how to make the silk in a spider's web, and they have put that gene into the goat. And now this goat The milk that they collect from it has large quantities of the goats. The goat's milk has the spider silk in it. Scientists are trying to figure out a good use for the spider silk because we found that it's extremely reliable product in terms of building. Spiders and their webs, from the small scale of a spider, a web is very, very strong. Not to us, because we can break it very easily. But if we were to take that property and make it into something very large, maybe a bridge, could we make bridges out of spider silk? I don't know. We got a bunch of biochemical engineers that are probably working on this right now. And moving over to page 35, let's talk about gene therapy. So there is a way that we can remove cells from our bone marrow. So actually having to go into your bone. There's bone marrow in there. Those of you that are in anatomy, this should be familiar to you. And remove stem cells from the bone marrow. In those cells, there is, if you're doing this process on a person, there must be some gene in there that's not working very good. And if they could remove the gene or reverse the gene in some way to make it a normal functioning gene, and then put that normal gene back into the cell, and then return the genetically engineers back to that patient, the thought is that that perfect cell, or the normal engineered cell, would then make more copies of itself, and then that's the cell that would be dominantly in the bone marrow, and not the one that is that was defective. And moving on to our last page of notes, page 36, let's talk a little bit about cloning. There's a big debate out there. Can we clone people? Should we be cloning people? Is it even possible? Well, we started with a sheep. We started with a sheep. The sheep's name was Dolly. They did this in the mid-1990s, was the first time it was actually successful. The process didn't work on the first time. They tried about 265 times before it actually worked. So there was a bunch of times where it was epically failing. But the basic premises behind how it works is this. They took a cell from any sheep, and it's a regular body cell, so they could have scraped it off of his ear, or they could have taken a cheek swab out of his mouth. Wherever the cell came from, it doesn't matter, but they did take the DNA out of that cell. So they got rid of the cell membrane, they got rid of all the cytoplasm and all the organelles, and they just took the DNA out of the cell. From a second sheep, they took the egg out of that. They took an egg out of the ovary out of that sheep, and they removed the DNA from that egg. So the egg is still there, but it has no nucleus, it has no DNA in there. So they took the DNA from the other sheep and they put it inside the egg. Now the reason why they had to use an egg is because an egg is a type of a cell. It's a gamete, so it's ready to become an embryo. It's ready to start the development of a new organism. So that's why they needed to use that type of a cell from the second sheep. That egg, once it had the DNA in there, was then stimulated to start dividing into an embryo. Once the embryo was big enough, they put that embryo and inserted it into the uterus of another sheep. And after a certain period of time, I don't know what the gestation period of sheep is, it's less than humans, so less than 9 months, the sheep then gave birth to a baby sheep. Now, this sheep is a clone of sheep A, because that's where the DNA came from. It's not related to sheep B at all. It's not related to sheep C at all. It is a clone of wherever the DNA came from. We have tried this with other organisms. We've tried it with, I know we've tried it with horses. Here's an example of we've tried it with cats. This is the original cat named Rainbow, and then the clone is called CC for copycat. They are genetically identical. Now, if you look carefully, though, they don't look exactly the same. There are some color differences between the cats. But just like with identical twins in humans, although they may have the exact same DNA, there are probably some physical differences about the shape of their face or their height or the way they stand up. There's probably some things about them that look a little bit different, and that's what has happened with the cats here too. They don't always come out looking completely identical because they were not in the same uterus. The mother carrying them may have been exposed to different foods or different environmental conditions. So there are some environmental factors that have an impact on their phenotype. But in terms of genetics, their DNA is exactly the same. So that concludes our notes. Your molecular genetics packet should be totally complete, and I hope this helps.